All right, so we have a really fun afternoon planned ahead as we continue with our program to look at regional food stories from around the world. I'm so excited to hear about the work taking place at the incredible centers of excellence, universities, public health institutions, and others on each continent that are championing food quality research, capacity strengthening efforts, and the translation of science for impact in their regional landscapes. The first part of these regional stories will feature the Centers of Excellence from the Americas and Oceania. So I'd like to invite Andres Jaramillo Botero, a professor at the Javerian University and I messed that up, you'll correct me, and director of multi-scale modeling and simulation at the California Institute of Technology. We also have Natalia Vasquez Manieres, a researcher in medical sciences at the National Institute of Medical Sciences and Nutrition at Salvador Zubarrayan. We also have Justin Ziegel, an associate professor at the Genome Center at UC Davis, and Vincent Lal, who is the laboratory manager at the Institute of Applied Sciences at the University of the South Pacific. Please welcome them. Bigger round of applause, everyone. We don't want to get tired after lunch. Okay. I'm so glad that you all are here. It's great to be connected to all of you. So we're just going to go through each of you each have 5 minutes to present. So I'm going to start with Andres. Can you go first?
Sure. Thank you, Danielle. So over the past 20, 25 years, I've been working on the fundamental understanding of material properties from their constitutive elements. And that means, for me, atoms, basically atoms, okay? So I see atoms as building blocks for everything. We might, in the PTFI, see molecules as building blocks, but in the real sense, atoms end up doing the chemistry, doing the interactions that really define us and define our health. So you might say I have a very simplistic view of food. For me, food is what we eat. It's extremely conditioned by our genes and by the environment. So the bottom line is we need to eat atoms that sustain our existence, our individual existence. I don't see a single diet that can be good for everyone. So in that sense, of course, there's also taste and habits, and they usually preempt our rationale. For me, it's pizzas, right? So we cannot, at this point, unfortunately, control matter at the atomic scale arbitrarily in 3 dimensions. So what I'm going to try and tell you about today is what we're doing at the Center of Excellence in Colombia, at the OMICAS, at the Pontificia Universidad Javeriana.
Thank you. Sorry.
Sorry for the pronunciation. It's
sorry.
It's hard. That's the PTFI Center of Excellence in Colombia. So, and I will try to give you a roundup of what we're doing with a very complicated diagram that you see on the screens. So we start by selecting and obtaining the food samples. Of course, we look for diversity. Procurement is an issue in these countries for different reasons. We need to identify the varietal sources. We need to identify the harvesting protocols. We need to identify how they collect the samples and how these are, eventually commercialized. Most of what happens with, you'll see in a minute, with fruit, for example, is very informal. So it's informal economies. And the question is, how do we trace all this back to the genes and the varieties that were used to produce these products? So procurement is a heavy problem. In some cases, we do extraction both at the pulp level, at the seed level, and of course the skin. It is important, and I'll show you an example that has given us really different results in terms of the composition. We then isolate the individual biomolecules. I think we've had several talks discussing this to look at the biocompounds, minerals, micronutrients, macronutrients, and so forth. But think of this. all of these components we are picturing here as a three-dimensional puzzle. They're all self-assembled by the organism, right? And we're trying to solve this three-dimensional puzzle of millions of components by then reducing it to probably 2D representations, and that seems to complicate things. So the result from this 3D structure is then, or 3D composition is then reduced to 2D. We have to deconvolute the individual components. Right now, for example, in metabolomics, we produce something called a mass spec that gives you the mass components of those individual biomolecules that you have in there. And then you need to decipher somehow what is the three-dimensional structure associated with that particular mass set. That's easier said than done. So we have to use, supercomputing and ultra computing to actually get to understand, and especially in the black matter that we've been talking about. Now, eventually that structure determination is what plays a pivotal role in determining or deciphering the chemical makeup and the biofunction of these identified molecules. So for us, it's critical that step going from that 2D to the 3D structure, because at the Institute at IOMICAS, we're looking to, once we have these compounds, understand how each individual compound is interacting with the tissues in a human being. And we can do cell line analysis, taking each biocompound and looking at how it interacts with lung tissues, with cardiac tissue, and so forth.
Wow.
It might be very simplistic, but that's the first step. Eventually, we need to do mixtures of these compounds and see how the synergy between them generates some other effects, both health-wise and in other terms. Now, for us, it's extremely important to then start playing another game, and that game is how can we put together new puzzles to make diets personalized, to make nutrition personalized, health personalized. And eventually, we can also use the output of PTFI, including what's being fed into this database. We can use all that for other purposes, industrial applications. We can extract surfactants. We can extract a whole bunch of molecules that have significant relevance in the industry. And that is part of our interest as well. So now let me drive your attention to the bottom of the screen where we have the demo projects. And I'd like to tell you a little bit briefly about what we're doing there. So in an effort to, and this, by the way, is with the help of Verso, Colorado State, and of course a team of very qualified personnel at IOMICAS. I'm just going to give a shout out to Gabriel, Juliana, Lina, and those who are in the front lines, including our support staff. So in an effort to highlight the importance of the validating the traditional cultural knowledge against scientific research, we have been looking at edible fruit biodiversity in Colombia. So Colombia has right now about 95 different fruit species that are commercialized on a daily basis through these informal channels, okay? Now, if I give you a single example of what we found, we looked at chontaduro, and I think Maya mentioned chontaduro. It's a small coconut, comes from a palm tree. It has a pulp, it has a seed, and it has a thick skin. Now, it is commercialized openly and loudly as an aphrodisiac. So out of these 95 exotic fruits, half of them have these very super food properties that are very interesting to see in the real world and how they sell them in the street. So we went ahead and we started doing metabolomics and we started doing fatty acid analysis and so forth. And lo and behold, what we find is, yes, there are some secondary metabolites and other compounds in there that do have a direct biological association with vasorelaxation of localized, tissues. They have, they're promoters of our hormonal synthesis. Yes, we've found those, but not in the pulp. They're in the seed. Nobody eats the seeds. Nobody's consuming the seed, right? So And we also find in the pulp omega-9s, which are, unsaturated, monounsaturated fatty acids. They're very healthy, actually, and that is nowhere to be promoted. Nobody talks about these, but we could be using that information as well to produce other foods based on chontaduro. Now, these fatty 9 acids have, very good prevention of risk of hypertension and cardiovascular diseases, so we could be using them for that. Now, in the second slot of the demo projects, I have grains, and that's because
I'm so sorry, Andres. I'm pretty sure that I told everyone they had 5 minutes. I'm pretty sure I didn't mispronounce that, although I mispronounce a lot of things. So I'm going to have to cut you off. I apologize.
Okay, so can you give me 10 seconds?
Sure.
Okay, so with beans, we're analyzing a set of accessions from the Future Seeds gene bank at CIAT, which has about 39,000 accessions. And we found very interesting things comparing lima beans against common beans and other types of beans so forth. So I think what we're heading towards at this point is really looking at how we leverage from these PTFI toolset to actually extend the application range that we can do in our countries and in other countries, of course. Thank you.
Round of applause for Andres, please. Natalia, I know you can see the time clock right there. Please go ahead. I'm excited to hear from you.
Okay. So, good afternoon, everybody. Today, I would like to talk to you and share with you the story of 2 interconnected studies that, in hand with PTFI technology, will help us understand better the Mexican food composition, its biomarkers of intake, and the relationship or how we may use them to better understand human health. We've selected different foods from our country that are not only important for our cultural identity, but also as for our status as agricultural producers. These foods are, the first 6 foods are mango, avocado, tortilla, guayaba, amaranto, and nopal. Simultaneously, it's important to remember that part of our mission is to bring back to the Mexican table different foods that have been underrepresented in our diets in order to bring biodiversity back. These 2 projects, BMX and MAPAMX, aim at profiling Mexican foods and discovering their biomarkers of intake. BMX is a randomized controlled trial that will develop, along with PTFI technology, the profiling of urine and serum in order for us to understand what are those compounds specific for each food that we may then use to correct the information that we get from dietary assessment methods. You see, these dietary assessment methods we know carry bias because of their retrospective nature. Therefore, when we want to go back and look into what is it that people are eating and connect them to human health, things become quite challenging. You can only imagine what happens when we want to go to specific populations. Think of the elderly, think of the children. How can we actually do a better job at understanding what are the connections between our dietary intake and different sicknesses, such as diabetes, heart disease, or other cardiovascular conditions? MAPAMX, on the other hand, has a mission to profile these different foods. Besides the 60, with the 6, we're looking at 48 others that are also deeply in our Mexican culture. We will look at things like chia seeds, the maguey pad, as well as different quelites. The information of these foods will be contributing into the PTFI database, and this will give us the chance to understand how is it that foods compare to other foods integrated in dietary patterns that have already been proven to be health benefits for humans. Our Center of Excellence will concentrate in the analysis of lipids, but will also leverage on other of our partners in order to use other technologies, such as metabolomics. Furthermore, these 2 projects, BMX and MAPAMX, are at the core of our Good Food Fellow program. With this, we hope not only to participate in science, but to create as well new individuals with a different way to look at science. In conclusion, BMX and MAPAMX, along with PTFI ecosystem, holds the promise of transforming and understanding the Mexican diet and its health implications. Thank you.
Thank you, Natalia. Well done. Well done. Thank you so much. So exciting, this work. Justin, I'd love to hear from you now.
Wonderful. And I think, I just want to say, having been in part of these conversations from very early on, I just want to thank you again, Roy, for your leadership and your vision and the team you've brought together, particularly with Selena and John and the broader team around it. They've done an incredible job taking a nascent concept into a reality. So just thank you. I'm going to give you applause. And I want to echo, I think it's really critical, and I'm just going to continue echoing this. We have to move from quantity to quality. We have done an incredible job at generating a massive amount of food and delivering it to the world in an incredible way over the last 30 years, such that today we are now living for the first time in human history in an era where obesity is a bigger problem than hunger. That was released by the World Health Organization. That's the middle diagram here a couple weeks ago in the Lancet. There are now more people, including in the global south, that are living in an obese state as opposed to a state of hunger. That has never happened in human history before. And the reason for that is the quantity over quality. So we're now in a ripe time to start changing that. But the beauty, as we've heard and alluded to, this isn't the first time we've done this type of thing and try to introduce quality into the diet. I can have a conversation with my grandmother about the quality of her food today. It's not a totally foreign concept. For example, if I give her a food and I say, hey, this is full of vitamins, my grandma would probably ask me, which ones? We would have a conversation about the molecular composition of her food, of all those components at the atomistic level. And that has been democratized, and incentives have been aligned around the producers, the manufacturers, the distributors, and the consumers to deliver that. So we've done that before, but then we stopped at the micronutrients. We never moved on to the macros, as we heard before. We thought we knew it all in the 60s, and we literally stopped. So we said, hey, proteins, fats, lipids, fibers, whatever, right? That's just this macro. It's 99% of the food. Like, that's crazy to say, this 1% is all we really care about the composition. A protein's a protein. Protein is the second most important macronutrient relative to water. That's it. That's the only thing more important than protein is water. And number 2 is protein. If I ask my grandma, or I guarantee any one of you in this audience, hey, I got this food. It's full of protein. You might ask me, where's it from? You will not ask me which ones. You will not ask me about that molecular composition. And so that's what we're doing. We're trying to figure out how do we get that down to that molecular composition, because it's so important. On the data showing I'm right, you can now ask me which ones. For the first time, we have examples of a pea and a pork burger, and that is the molecular compositions. Those are distinct proteins. Those are not 20 grams of protein each. It's 20 grams of protein, but they are produced of visilins versus myosins and actins, which have different functionalities. So if you look at the plot in the middle, those are 2 different people eating those. We have 4 different burgers in this case. And one of those people is responding really well. It has high bioavailability for the pea protein. And one of those people has high bioavailability for the pork protein. We're not all the same, as we alluded to earlier. But we don't have to stop there, because the PTFI, we now understand not only are there differences between us, we understand molecular compositions of what's in there. So we're now using AI tools to go in and figure out what specific components can we digest and not digest. And I don't have to go to Roy and say, hey, Roy, unfortunately, you're a pork responder. You're going to have to only eat pork if you care about your health. My guess is you're not going to be thrilled about that. But because of the understanding of that molecular composition, we now have actually created the tools and technologies that I have early data showing that I can take that pea burger and make it respond as good or better than the pork burger. So Roy doesn't have to end up eating the pork burger. He can eat the pea burger, and it will end up being better for him because we understand the composition, we understand the recalcitrant proteins, and we can deploy technologies to unlock it. So I'm incredibly excited about what the future lies as we unlock this black box and are able to deploy tools and technologies to get there.
Thank you so much, Justin. And thank you for bringing up that point. Please give him a round of applause. We really have to be better at nourishing folks rather than just filling them up. So thank you for reiterating that. Vincent, you're up next.
Hi, good afternoon. Thank you for this opportunity. I come from a region of Oceania and representing our Pacific Island countries. And the work we are doing is on biochemical diversity analysis of different varieties of indigenous and underutilized crops from the South Pacific. Now that is a very pristine location. It's idyllic. Everybody's bucket list probably almost to go to the South Pacific. But this is a good changeover from the talks because you see a lot of people. We are topping the charts on NCDs as well. So in terms of climate change also, you know, Pacific Islands. So these are the realities of the region as well. And I think the work, the periodic table of the food initiative, the opportunity that it provides to a region that is sometimes so isolated and disconnected is itself a big achievement, I think. But moving from there, the work that we are doing, I have my boss here too, Dr. Iswa, mostly involved in capacity building and training work. So that comes naturally to us. I come from the University of the South Pacific, the Institute of Applied Sciences. The university is co-owned by 12 Pacific Island nations. So we hold the mandate for the island countries in a way, in terms of research and development. And food is very important for Pacific Islanders. It is gone wrong, in the wrong direction. And a lot of people are suffering NCDs, and it has implications for communities. People have very low life expectancies. So the work that we are trying to do with the PTFI is to go back to our local food systems. There is a big disconnect. And I think Pacific Islanders, just like many parts of the world, are influenced by a lot of simple nutrition lacking diet, sort of high energy, less nutrient rich diet. And you see from the image there, very beautiful foods. And these are the foods that we want them people to go back to eat more. And I think how they will go back to it is when they get to know what is in the food and what these foods contain. Why should they connect again to these foods and how it can heal them, heal those communities. I think it's an opportunity that we have through the PTFI to change the life of Pacific communities, and through data. And we have a lot of partners that we work with. For example, we have Robert Oliver here with us, Pacific Island Food Revolution, that does a lot of television work. So not that the data that we work on, for example, the last food composition tables for the Pacific Islands was in 2004. 20 years of gap. What does it translate into? People don't understand what they're eating, what's in their food, what's in Pacific Island food. So these are implications on a regional scale, I think, and building capacity, training the next generation of local scientists, able to generate data yourself. This is an opportunity that the PTFI has brought to us, to our institute, and generally to go back to doing research that is meaningful, generating data that is meaningful, and connecting to people and telling them about their own food. I think that would be the most important thing that we can do through this research, and we are very grateful for the opportunity the PTFI has given us. Thank you.
Thank you so much, Vincent. I love that you brought up reigniting this interest in indigenous and traditional foods, and that your slide showed fish, and you mentioned Chef Oliver and this need for more of an emphasis on blue foods. And I forgot to thank fishers when I was thanking farmers and farm workers. But please give this panel a round of applause for their incredible work. Thank you so much.